also nice thing about those two laws is the fact that I can treat each allele independently of each other, and so I can use the laws of probability to predict the likelihood of the outcome of a particular event. So not only can I use my Punnett squares as a way to help, but then I can also look at 10 traits, and rather than having to do this ginormous Punnett square, I can take each trait individually and use the rule of multiplication to multiply those individual alleles together, or individual letters together. And that multiplication, that math, that statistics that I can use, tells me, all right, if I want to know what the probability of having a dominant A, B, C, D, E, and F is, I don't have to do a huge Punnett square for all of those. I can take the probability based on what my parents in my cross are passing on for their A allele and get that probability, and what they're going to pass on for the B allele and get that probability, and get C and that probability, so on and so forth, and multiply each of those probabilities for each of those individual alleles together to get the total possibility of getting dominant across the board. So let's look at monohybrid crosses for a minute, because really when it comes down to all of it, because I can treat each trait individually because of the law of segregation and because of the law of independent assortment, really at the end of the day I don't have to do more than a monohybrid cross, because I can come back and use this. So when we talk about monohybrid crosses, this is where we're talking about mating two individuals together, two parents together, and only looking at one trait at a time. So something like what Mendel did with mating his different pea plants, and he looked at mating a plant with yellow peas to one that had green peas. So all we're caring about in this particular cross are what color the peas are. All right, and here's where I can use those Punnett squares to help predict the outcome. So here are those Punnett squares, and these are perhaps ones you've seen before because they're relatively simplistic. You're just looking at one trait and two parents and what is the predicted outcome of that particular trait from these two parents. And keep in mind that your Punnett squares really are only there to be utilized as a tool to predict the probability. Because when you go through a mating event, the egg that the mom gives and the sperm that the dad gives are going to be randomly selected from all of the eggs and sperm that they could possibly be making from that particular set of meiotic events. Okay, so at the end of meiosis, remember that dad makes 4 different sperm, mom makes 1 mature egg, but 4 polar bodies, and so the genetic material that they happen to get into those sex cells, and then that particular egg and sperm that get fertilized, it's all random. Okay, so the outcome of an actual mating event may look different from the actual probabilities we're using and we're getting from a Punnett square, especially if you're talking about an outcome like humans who only have 1 offspring at a shot, or from even cats or dogs who have a small litter. The only way that you're going to get ratios of offspring that look much closer to the probabilities we get from a Punnett square are from organisms that make lots and lots of offspring, not only in a single mating event, but also over their lifetime. So if you're talking something like fruit flies or plants who could have lots and lots and lots of offspring over their lifetime, then maybe you're going to start looking at getting probabilities that are a lot closer to than what you're predicting with a Punnett square. But what the Punnett square can do is at least tell you that these are the possible options for a particular cross from these parents. So it's not a total waste. So how can we construct a Punnett square? Well, you've got a couple of options. You can do just the normal 4 boxes, which is probably what I'm guessing most of you will do, or I've seen it where they show you how to make the tic-tac-toes. Okay, so let's go through and use that as an example. So your parent A, all right, whoever your one parent is, their alleles are going to go across the top. Typically, this is your male parent if you happen to know that information. And in this case, this parent is heterozygous for this B allele that we're tracing. Okay, so big B usually goes first, little b goes second. Second parent goes across the side, and generally this is usually mom's information, again, if you know who's providing what. And so this particular parent happens to be homozygous recessive, so two little b's, right? Then you go and you pair them down the column and row. So we've got this big B coming here and here, little b's going across, little b's going down here, and these going across as well. Okay, and so these are your offspring. These are parent 1 and parent 2. And so we can now use that Punnett square information to predict the genotypes and phenotypes of the offspring that we've created from this particular cross. So remember that your genotypes are the alleles, the actual letters that the organism carries. So in this particular cross, I could have had a homozygous dominant, so two big B's. Could have had a heterozygous, big B, little b, or I could have had a homozygous recessive, little b, little b. Those are all the potential options from this cross for this particular trait. So we're not even necessarily in this exact scenario taking into consideration what the parents are yet. Okay, that's just the potential possibilities for genotypes. We can also use the Punnett squares to predict your phenotypic ratios, so what the organism is actually going to look like. So in this particular scenario, we're being told that black is dominant. So homozygous dominant, two big B's, or heterozygous, big B, little b, is going to produce an offspring who has the phenotypic expression of this black trait. Homozygous recessive, so two little b's, is white. And so we can use those understandings of what the potential genotypes, what the potential phenotypes will be to give us the ratios from this cross. So having a parent who is heterozygous mated with a parent who is homozygous recessive, I'm going to end up with two heterozygous individuals to two homozygous recessives. So a ratio of 2 to 2, or if we think of them kind of like fractions, we can reduce them to 1 to 1. So one heterozygote to one homozygous recessive. Phenotypes, well, heterozygotes are going to be black because they have a dominant allele. So I'm going to get two of them from this cross. And I'm also going to get two whites because white is recessive. So again, it's another ratio of 2 black to 2 white, or I can reduce it to 1 black to 1 white. Now, depending on what the genotypes of the parents are that you're crossing together, you can get different genotypic and phenotypic ratios, right? It's all going to be dependent on what the parents can pass on. So if we use a similar idea, but now I made two heterozygote parents together, both showing the dominant trait in this particular situation, that dominant trait happens to be purple, recessive is white. Both parents are heterozygous, so they have a big B, little b, okay? And when we cross them now, I end up with one homozygous dominant to two heterozygotes to one homozygous recessive, or a 1 to 2 to 1 genotypic ratio. Phenotypically, though, I end up with 3 dominant, so 3 purple to 1 white, or a 3 to 1 ratio, which is kind of consistent with that traditional Mendelian genetics, something that we would generally look for to say, all right, if I make two heterozygotes together, I should get that 3 to 1 traditional ratio. If I don't, then we can start talking about some potential other options of inheritance patterns, which we'll get into next week. But what if I don't know the genotype of the parent? If this is a traditional Mendelian cross, my dominant parent could be homozygous dominant, so 2 capital letters, but they could also be heterozygous, and there's no way for me to know that simply by looking at them. So one of the things that geneticists can do to try to sort of backtrack and figure out what the genotype of a particular dominant parent is to do what we call a test cross. And these test crosses are going to take that dominant parent, the one that we don't know, is it homozygous dominant or heterozygous, and we're going to mate that dominant parent with a parent who's recessive. Because we know that if that parent is recessive, then they have to be homozygous for it. They have to have 2 little letters. And so we can mate these individuals together and look at their offspring. The ratios that we get from their offspring will tell us whether or not that dominant parent was homozygous dominant, 2 capitals, or heterozygous, a capital and a lowercase. So how does it work? So here's an example going back to Mendel's pea plants again. If I look at this parent, okay, it's purple. Purple's dominant. And so they can be homozygous dominant or they can be heterozygous. I'm going to mate that questionable dominant parent with a recessive one. In order to be recessive, you have to have 2 lowercase letters. So we mate these 2 individuals together and we look at what happens in their offspring. So if I mate these 2 parents together and this parent was homozygous dominant, then all of my offspring are going to be heterozygous, and so they're all going to show purple. But if I happen to get a white offspring out of this cross, then I know that this parent had to be heterozygous, because the only way that this works is that that parent had to have a lowercase p to pass on in order to get the white ones, because one lowercase came from here, the other lowercase came from here to get us this. So these test crosses are really convenient as a way for us to kind of predict some ratios. So let's assume that my parents were in fact true breeding parents. So if I'm true breeding, it's kind of the same thing as saying, right, purebred, and that means that they are homozygous for their trait. So this parent would be homozygous dominant, so 2 capitals. This one would be homozygous recessive, so 2 lowercase. If I use them as my parent generation or my P generation and mate them together, my first generation of offspring are all going to look dominant if this is traditional Mendelian genetics. And so that's what we talk about as your F1 generation or your first filial generation, F1. Okay? Filial meaning family. And so your first generation in this type of cross, they're all going to be hybrids or heterozygotes, right? If I take 2 of these individuals from this generation and mate them together, so 2 heterozygotes that I mate together, then in my second generation, or my F2 generation, that's where I get that traditional 3-to-1 phenotypic ratio, 3 dominant to 1 recessive, and my genotypic ratio is going to be 1 homozygous dominant, 2 heterozygotes to 1 homozygous recessive. And so all of these different ratios, understanding how Mendelian's genetics work, allow us to use these test crosses to backtrack and understand what the genotypes of these dominant parents are, because I can't just look at them and know. Before we move on to the next topic, there's one more thing I wanted to just point out, because we're going to sort of move away from Mendelian genetics and start getting into some exceptions to what Mendel saw with his pea plants. Before we do that, though, keep in mind that we're talking about genes thus far that are autosomal, meaning that these are on chromosomes that are not your sex chromosomes, in which case you need 2 copies of that gene, assuming you're talking about some sort of recessive trait, right? So if we talk about some sort of disease like cystic fibrosis you see here in the diagram, you need 2 recessive characteristics, 2 mutated traits in order to show that particular disease, because this is a recessively inherited trait. That being said, not every disease or disorder necessarily has to be inherited recessively. Overarchingly, they are. And if you think about the reason from that, it really goes back to evolution. So if I'm recessive, that means I have to inherit 2 bad copies. So the chance of me having that is about 25% if I assume 2 heterozygous parents who don't show the trait and are healthy. But it can still be a dominantly inherited characteristic. And the reason being is that oftentimes those dominantly inherited traits that are sort of disease causing or not as beneficial to the population will happen when that disease's symptoms show up post reproduction, right? So go back to evolution, everything comes down to reproducing. If I am capable of reproducing, then that trait gets passed on. If I can't, then it starts to get weeded out of the population. So for a lot of things like cystic fibrosis, it's recessively inherited partly because that trait, beyond the way that its genetics work, also isn't very common in the population either because it often kills its individuals by their mid-20s. So that's not really a huge opportunity for them to produce copious amounts of offspring. But if I talk about something like Huntington's disease, that particular disease doesn't really progress or show symptoms until somebody's 40 or 50, at which point that trait is actually dominantly inherited and still maintains its presence in the population because it's not showing symptoms until that person is probably past reproductive age. So, what's my point in telling you this? Just be aware that recessive doesn't necessarily mean uncommon. Dominant also doesn't necessarily mean common. These are just terms that we're using as the way to describe the way traits are inherited. It's based on their genetics, not necessarily about how they function in a human being. In fact, go look at your hands right now. Webbing between your fingers is actually a dominant trait, and yet I'm going to guess that most of us are sitting there without webs between our fingers because it's not really very adaptive for our environment. Another point I want to make here, though, before we kind of close this chapter of genetics, is that if a disease or if a disorder or if any trait you want to talk about is in fact recessively inherited, then anybody who is heterozygous is what we would consider a carrier, meaning they don't show the trait, but they carry around the instructions to potentially show that disorder or show that trait. So here's my heterozygous individuals. These guys would be considered carriers. Why? Because they're carrying around that cystic fibrosis gene, that cystic fibrosis allele, but they themselves are not actually showing the trait. You can't have a carrier who is carrying something for for a dominant trait because they don't carry it. They will just show it. Does that make sense? So heterozygotes can be carriers because I don't show the trait. This only works if the trait is recessively inherited. If it's dominantly inherited, there is no such thing as a carrier for it because you just show it. You know you have it. So hopefully that makes sense.